Thank you very much, Dr. Zadnik, for this nice introduction. Also, I would like to thank very much the sponsors of this lecture series, Dr. Richard and Leonora Hill. And without their sponsoring, I wouldn't be here for sure, but I wouldn't be here either if not Don Moody would have been my host here, mostly, although the others are also my host, but Don was in Tübingen in 1992 for a 3-week or 2-week visit, and this connection lasted for the last 30 or what? How many years is that? I don't know. Yeah, long enough. Okay. Okay. My work that I'm reporting today is to 80% made recently in Basel. I got a guest professorship in Basel in addition to my senior professorship in Tübingen. And so I travel back and forth between Switzerland and Germany, but in Basel we have a small group, which is shown on the left bottom here of the slide, Barbara Swerczak and a PhD student from Paris, which recently joined us, Leon Garcia. And most of the work is from there, but I will start with old work that is known by most of you, but you may not have seen all the pictures and connections that I believe I found over time. So for instance, if you would like to study emmetropization, it is great if you have an animal which emmetropizes very obviously. And at the time in the 90s, I had a person at the Max Planck Institute, Hermann Wagner, who had studied only vision in barn owls. And I was doing some work with him together. And the interesting thing is in these barn owls that they keep their eyes covered. The lids are fused until 13 days of hatching, which means the eye grows for quite a while without any feedback. And now if you refract these barn owls, when they first open their eyes, they really have a lot of refractive errors. So this is the infrared photorefraction, which you know where you, if it's bright in the bottom of the pupil, they are myopic. If it's bright in the top, they are hyperopic and so on. And this is one animal that we followed here for 16 to, from day 16 to 45. And you can see how nicely myopia disappears over the 3-week period. At the end, this optics is just perfect. And you can see here another owl that started out hyperopic. It really overshot the 0 line and then got back to the best refraction that they can have, which may be between 0 and 1 or so. And you can see that both animals really had a perfect feedback loop behavior here. They ended up in the correct refractive range. How does this work? This was a long question, and initially it was assumed to be a genetic control, but this has to be given up because it's just so precise that genes are unlikely to be able to do this. And it started all in the 70s, as you may know, that Thorsten Wiesel and Elio Raviola found by accident that eyes of monkeys who had lead fusion to study amblyopia, actually, they got very long. And at the same time, only 1 year later, actually, Josh Wallman did this first experiment in chickens, putting these diffusers in front of the eye. And this ended up as a science paper in 1978, showing for the first time that a modest disturbing of the retinal image already has a big effect on eye growth. And I show you here on the right some studies in chickens that were done in the 90s. Actually, one eye was occluded with this occluder, as you can see, and the other eye was having normal vision. And you can see that these are data from 7 chickens. You can see that one chicken gets very myopic in a week, so -26 or something, and the others less so. But what is striking is the other eye didn't care really. Eye growth on the other eye was normal, and so it was strictly a change that happened in the treated eye. So it's not coupled in this case, but it's true also for monkeys. Now, 2 other important discoveries came up there. This was that you don't need the optic nerve to do these experiments at all. If you cut the optic nerve in a chicken, which wasn't done by me, by rather Chris Wilsett and David Troilo at the time, you still get this ocular elongation. And we found, as Josh Wallman actually found here, this was also Science 1987, that if you take half occluders, you get only half eye growth in the areas that were having poor vision. I should say that this science paper saved my career because at the time I was applying for money from the German Research Council to do studies in myopia, and they said this is an esoteric topic that can't be scientifically approached. Just at this time, this paper came out, and then it saved me, and they believe now that it's worth to be studied. Yeah, and at the same time, I was working with Howie Howland in Cornell University, and I prepared some of these chickens, and I found this wonderful optics, this clear lens, and everything so exciting, like a real lens. And so I thought we should add some additional lens and see what the eye is saying to it. And so I spent a lot of time making these hoods, as you may remember. We no longer use hoods. It's so tedious to get them in all these sizes because eye grows pretty fast in the chick. So every 3 days a new hood. And we had a huge selection for many chickens. So, and I did them all myself. So I spent almost a year to make just these leather hoods. But anyway, the experiment worked extremely well. So even if you put a negative lens in front of the eye in 3 or 4 days, its lens was fully compensated by increasing axial eye growth, and the plus lens actually eye growth stopped. And because the anterior segment continued to grow, the focal lengths got longer and they got hyperopic from that. So there was a bi-directional response of the eye, which was the new thing, because deprivation myopia is open loop, and no matter how the eye grows, it always will be out of focus. Now the big question is, how does the retina separate or distinguish whether this is just deprivation or there's some focus on top of it that is actually should force the eye to go in the other direction? And this is something that we solved only in the last year, I guess. I hope we solved it. But it took 30 years to solve it, and you will see later. I first would like to show that this defocus detector or sign of defocus detector is extremely stable and works in very poor image quality. Because the experiments that we did this by Sigrid Dieder, which you see not really well here, but maybe you see it. We put the chickens in the center of a big drum, and there they had only one viewing distance, which is about 3 diopters. But then we added lenses so that their far point was either 12 diopters in front of the wall. So in this case, you take strong positive lenses, or behind the wall, which meant that the image was very poor in both cases. And you would have expected that all eyes become myopic because they develop deprivation myopia. But you can see already here, the eyes could perfectly detect that there's also a plus defocus, and they shouldn't get longer, and they didn't. And another experiment by the Wallman group actually is even more convincing, I think. What they did, they gave chickens plus lenses. In this case, the eye gets shorter, as you can see here, and the refraction was getting more hyperopic with plus lenses, as you expect. But what they did now, they added Bangada foils. These are diffusers on top of these lenses, basically let deprivation myopia compete with the plus lens and see what happens. So basically what they would expect probably is one drives the eye to get longer, the other drives the eye to get shorter. So you would expect no change at all. But the result was very different. What came out is that with the plus lens, all these eyes became shorter and hyperopic, despite that there was considerable amount of deprivation. So this is, for instance, a Bangada foil with 0.1 vision, 10% only of vision, and still the eyes got definitely shorter. So the mechanism that inhibited eye growth was much stronger. Only if you make the diffusers completely diffuse, so there was no image getting through, then they started to become myopic like you expect. So this was an experiment that showed that the inhibitory signal for eye growth is extremely strong and stable. And now we can ask, do we now know how myopia really starts and why does it start? And well, we know based on these experiments, we can't tell. We don't know why are people, in particular, why does myopia not inhibit itself? Because a strong inhibitory signal is present, definitely. And the other thing is, you know, the long story about undercorrection. It never worked really well in myopes, and it didn't inhibit eye growth as you would have expected from a plus lens. Undercorrection is basically a plus lens. It doesn't work, and this is a unsolved question for a long time. But what we have to show is that this strong inhibitory signal is also generated in the human retina, and it's interesting that we may not be aware of this signal because we don't need it for vision or accommodation. And this is one thing about eye growth control. The retina does a number of things that the cortex doesn't know, and this is, for instance, controlling eye growth is done by the retina, and it's not all information that is used to control eye growth really transmitted to the visual cortex. So things happen there that we don't see, and I think this inhibitory signal is something we don't see. Okay, how can we test what this inhibitory signal does, or whether it actually is also present in human eyes? And so the idea here was we compare real optical defocus plus lens versus calculated spherical defocus that we impose in some movies. And this can be easily done actually if you don't care about aberrations, which we didn't in this case, just only on spherical defocus. You can see here if you defocus an eye or a distant light source, it will become a disk of light if it's out of focus. And you can see this nicely here for this Christmas tree that was photographed with positive defocus. And using this simple paradigm, just that each pixel is now no longer just only a point, but rather a disk, which can be calculated. The diameter of this disk can be perfectly easily calculated for a certain amount of defocus. This way we could simulate defocus in the retinal image by just filtering a movie. And this can be done in real time. You just have to read out all the pixels and take a circular area around it, averaging it in. And when we did this by calculating pupil size or measuring pupil size and all the distances, we could perfectly match the real modulation transfer function for plus defocus with the calculated one. And this one is without defocus. Interestingly, because these movies are on a big screen, they are not containing spatial frequencies higher than 10 cycles, which was also limited by the processing speed of the computer. But that all the differences are in the low spatial frequency range. And how the movies look like, you can see on this picture here on movies. This is unfiltered, so it's how the people saw normally the movies. But now here is how the movies looked like when people had a plus 3 diopter lens in front of their eye, which generated 2.5 diopter of defocus. And this is how the movie looked like when it was calculated with defocus. So these two things look pretty much the same to you, but one contains really real defocus and the other is only calculated. Now we want to measure what is the output of this. We need some measure to see whether the retina made a difference in looking at these two different versions of the movies. And you may know from Josh Wallman's studies already in 1995, and Chris Wilson was also working with him at the time, that if you put a positive lens in front of the eye, the choroid behind the retina gets thick in pretty short time, maybe a few hours up to a day or so. And Josh Wallman concluded, or this was the title of his paper at the time, that there is a third mechanism of focusing the eye, which is basically just making the choroid thick and moving the retina closer to the focal plane. This works actually quite nicely in chickens. 7 diopter can be compensated, but not in humans, because you get these responses in humans as well, only they account for maybe 0.05 or 0.06 diopters only. So it's not a really focusing mechanism, but it's a nice trick to measure what the retina is saying. And you may have seen that also in children, if they get positive lenses or negative lenses, the choroid responds by getting thicker or thinner within 1 hour. Yeah, the question now is studying these choroidal thickness changes. Is this relevant really? Can we predict eye growth if the choroid gets thicker, saying now we will have a less progression of myopia? Well, nobody believed it in the beginning, but I think the papers that come out in the last years make it more and more clear that choroidal thickening is linked to an inhibitory signal for eye growth. So you can nicely use it and test what the retina says to your presented stimulus. I have here a few slides from Michael Collins, who gave a talk online actually in February, and he collected also data for this question: is choroidal thickness change a predictor for future eye growth? First, he found or shows data here that the longer the eye, the thinner the choroid. This is well known. This is in children. Here you see that if the eye is longer, the choroid gets much thinner. So here it's 300 in emmetropic or normal length eyes, and it's only about 200 or less here, or 100 even in these myopic kids. Then the second thing he tested out is where are the changes in the choroid, and actually this is this red area. This is only the central 20 degrees or so are the biggest changes. Finally, what they also did, they gave autoK lenses here and tested how much the choroid changed. And in fact, it was shown that the more changes you can induce in the choroid within 1 month, the more was myopia subsequently inhibited over the following year. And this gives a really nice argument that thickening of the choroid is really an inhibitory signal or reflects an inhibitory signal. Well, in the LAMP study, you may know this big study from Hong Kong with many atropine-treated children, about 450 or so. They concluded also, if you give atropine and the choroid thickens, which it does, actually, we found this in chickens also, this is a great predictor for the efficiency of this atropine treatment. Now, what we measured actually is axial length. You can see here this red is the choroid, and if the choroid gets thicker, which happens within an hour or half an hour easily, the eye actually gets a little bit shorter. So just by taking the lens star, which most of you may know, you can already find out whether the retina responds or not to the stimulus that you give. And now the results of this experiment. If the people watch the movies with normal focus, the eyes all got a little bit changes over time in axial length, but neither myopes nor emmetropes showed a significant direction. There was a little bit more noise in the myopes, but it either went shorter or longer. Then if we low pass filter the movie, which we did here in the middle data, you can see that the eyes generally got longer. And this is kind of deprivation myopia because they looked at these low pass filtered movies, and this worked equally well in emmetropes and myopes. But now the interesting thing came when we used these positive lenses, because the myopes again took these positive lenses as if it would be deprivation and got longer in a similar fashion as if the movie was low pass filtered. But the interesting thing is here in the emmetropes got significantly longer eyes, not all of them as you see, but on average this was highly significant, which means in the emmetropic retina it is detected whether there's a plus defocus or not. And the interesting thing now is there's this difference between myopes and emmetropes. The myopic retina does not respond to real positive defocus, which means it does no longer generate an inhibitory signal for eye growth. The alternative interpretation could certainly be that the biochemical signaling cascade from the retina to the choroid and to the sclera is kind of deficient in myopes. This could be another explanation, which we tested in a very simple way. I show you. First, the hypothesis is either the retina does no longer detect positive defocus in myopes, or the positive defocus is still detected but doesn't transmit the signaling cascade to choroid and sclera. And so the test is quite simple. Do we have any other visual stimuli to make the choroid thicker? And would this also at least work in myopes? And I would like briefly to introduce the on-off system. Everybody knows about the on-off system in the visual system. In contrast to a camera which reads out each single pixel, the eye doesn't do this. It takes always a comparison to the neighbored pixels. And basically this is a very good trick to make this reduction in pixels that are 125 photoreceptors in the eye, but we have only 1 million cables to the brain. So this has to be compressed by down to 1%. So photoreceptor pooling and on-off systems is a great trick to reduce basically the information that can be transmitted to the optic nerve. And it also preserves high spatial frequencies. And I just wanted to show you this Adelson illusion. Maybe everybody knows the Adelson illusion. But this shows you clearly that in contrast to a camera which reads out each pixel, our visual system doesn't do it and cannot measure absolute brightnesses very well. So you all wouldn't think that this is much darker than this, but they are exactly same brightness, which tells you again that your retina locally doesn't do absolute brightness measurements. Only by comparison you get an idea. So what is the sense of all this? Earl Smith in 1996 was the first to test whether the eye grows differently if you switch off the off or on channel. Actually, what he did, he switched off the on channel in kittens by using APB, which is a drug that decouples the on bipolar cells from the photoreceptors. And what happened then is all these cats became quite hyperopic, which means if you switched one of the two off, then the system is basically no longer working very well. And when I read this, I was interested in seeing how much on and off do we have in our visual environment at all. So I wrote some software just to map out the on and off input strengths in photographs. And this is very simple. I took one pixel or took all the pixels one after the other. and compare whether the center pixel is brighter than the peripheral pixels, which would be an on stimulating position. If it is the other way around, it's off stimulating. And when I did this, I saw that in an eye, in a picture like we see all the time, there's actually quite a nice balance between on and off. Accidentally, I took this camera and directed it towards the wall, and there was some printed text on the wall. And then I suddenly saw that if you have regular text, the off stimulation is much stronger than the on stimulation. But if you invert the text, all this inverts as well. So you can selectively overstimulate the on or the off channel just by inverting the text contrast. And now the question was, what are the eyes doing with this? And when people read the text, dark text on bright, we found that their choroid became actually thinner. Basically, this caused myopia on the long term. And this is the way we read all the time. But if you invert the contrast, the choroids got thicker in all these people. You can see this were only 7 people from the lab, and it was extremely significant in these 7 people you can see here. So this is another way to make the choroid thick or thin, is reading the contrast like normal or inverted. And now the big question, is this working still in myopic people? And this was experiment done by Barbara and Basel. And actually, we found that if myopic people, you see here, if they read this text with inverted contrast, their eyes got still shorter and the choroid thicker, just as in emmetropes. These are emmetropes, myopes, pretty much the same. Only with the plus lens, there was this big difference. With the plus lens, the myopes got actually thinner choroids and longer eyes, where the emmetropes went in the other direction. So the difference is selectively sitting in the defocus, not in any other processing, and the signaling cascade is intact. And now I come to the next big point. We still don't know how does the retina find out whether it should generate an inhibitory signal or a growth signal. And even though the receptive field structures of the on-off systems are great to measure how well the focus is in the image, how many spatial frequencies we have, it still doesn't show any information about the sign of defocus. And you know, this is an idea that comes from Fincham in 1953, I think already, that if you have chromatic aberration in the optics, any optics has chromatic aberration almost. And the eye has this we have about 2.5 diopters of difference in focus in red and blue. If the retina would measure contrast in blue and red, this would be a really great signal to tell where the focus is. If red is in better focus, you know the eye is too long. If blue is in better focus, the eye is too short. So this would be a great stimulus. And we only have now to show that the eye growth really uses this stimulus. On the way to this, we found this paper. Actually, you may know it anyway, but if people are color deficient, they get less myopic on average, which means this color processing is definitely involved in emmetropization. This was a paper where David Edgson was on it and some others in IOVS about 2021, I think. But now it's getting funny. Then people, after they knew that color is important, they start many experiments. I only show very few of it. So one paper came out in 2022 in the fall by Thakur and Verkichala in India. They let people read in red light, green light, blue light. And what they found is really striking, which I found very unexpected. In blue light, their eyes became shorter and the choroid became thicker. This is shown here. While in red and green light, the eyes actually got longer in their experiments. In white light, there was no significant change. And this is something that industry liked, because blue light was always assumed to inhibit myopia, and this seemed to be along the line. But you may have seen these experiments in the tree shrew that were done by Tim Gorn in Tom Norton's lab. If you put these animals in red light, they actually don't emmetropize along the normal path. This is this black line here, but they always get hyperopic as soon as they are put in red light. So it's just the opposite of what was found in humans, that red and blue and red. So this is a confusing business, and I summarize this in this picture here. Actually, there's a complex pattern of how emmetropization responds to narrowband light. So monkeys in the red light get hyperopic, as opposed to chickens and guinea pigs, they get myopic. Then you saw this study here by Thakur that I showed in a minute. The blue light made the eye shorter, doesn't fit to the red light, makes it also shorter. What? And so there is a big mess here, actually. Monochromatic light is not really a consistent stimulus for emmetropization. The reason is if the retina would really use chromatic aberration, it has somehow to compare the red and the blue end of the spectrum. But if you cut out half of the spectrum, the other half is open loop. Basically, you make this system open loop in part if you cut out a certain part of the spectrum that is necessary. And so what you have to do, you have to stimulate both red and blue, but you have to provide different levels of focus in both sides. And this is what we did. If you look at a regular computer screen, you have a blue channel, green channel, and a red channel. If you look at the chromatic aberration function for humans, you can see there is a difference of about 1.3 diopters between the position of the red channel and the blue channel. So there is actually, even though you don't see it when you look at a movie or at the screen, the blue is always out of focus or the red is out of focus. So it's always not perfectly in focus everywhere. So what we did now, we used, again, the James Bond movies and filtered either the blue end of the spectrum in the movie or the red. And this is illustrated here. So if the blue is low pass filtered, the movie doesn't look perfectly sharp, but it's okay. The people usually didn't see it. If you filter the red end, then it looks worse because we have our high acuity in the green-red LM cone system. So the red filtered low pass filter is a little bit more difficult to watch, but people watch these movies, and yeah, the details about the filtering is something I can explain to somebody who wants to know it. But the result now, if you make the red low pass filtered and unsharp, this basically tells the retina that the eye is too short because it can see the blue in good focus, the red is out of focus, and this happens only if the eye is shorter. What you would expect now that the retina takes this as a signal to make the eye longer. And if you look at the change in axial length, in this case, really we observed in 20 subjects, 20 emmetropes and 15 myopes was the total. You read it there. That in this case, the eye became longer. But if you do the other thing, you make the blue out of focus and the red sharp, then the eyes responded in the other direction. And this is a kind of, it's hard to explain this with anything other than that chromatic aberration is really used to determine the sign of defocus. So I think this was, we believed that we found the sign of defocus detector. And I must say, accommodation was started for a long time with focusing the blue or the red. So this is not completely new, only this is the first time that it was shown for emmetropization. And the second big crazy thing is that myopes didn't respond at all. So they couldn't use chromatic aberration to extract the sign of defocus and cause this inhibitory signal. And I think this is something that is really needing more study. Something changed in the retina of the myope. Even though they had normal vision, they are just young myopes, not very myopic, between 1 and 3 or 4 diopters. And still, there's a basic function is gone, and this is detection of chromatic aberration. We have now two mechanisms of emmetropization. Actually, for a feedback loop, you need an inhibitory and a stimulatory branch. Always, otherwise it's not a feedback loop. And what we concluded finally is, no matter whether you take a negative lens or occluder, you always trigger this stimulating pathway. And this stimulating pathway is studied by most people. This pathway has a pretty slow response. It responds strongly to atropine, so atropine blocks it, pyrazine blocks it, dopamine. If dopamine is going down, this pathway is triggered to go faster. You can inhibit with bright light, you know, all this. This is probably completely independent of color. I think this is a pure luminance business. But there is another pathway which is scarcely studied, but the evidence is so clear that it is there. And this pathway does not care about atropine. So if you give a chick a high dose atropine, it compensates the plus lens without problem. And actually this mechanism is normally extremely stable, only it breaks down in myopes. And this is funny. This pathway also uses chromatic aberration, is color dependent. And I believe this is a pathway we have to study in the future because it's so strong and can inhibit eye growth in emmetropes so they don't become myopic. Excuse me. Anyway, If this pathway doesn't work, the human myopic eye is open loop. It grows just without inhibition. And I think this is what the case is in myop. And I have two conclusions. I hope you still live. How long can I talk? Quarter past or so? Yeah, okay. So the conclusions for this part is the emmetropic retina generates a perfect growth inhibiting signal. Keep the eye emmetropic, it never grows too much, but this signal is using chromatic aberration and is only present in the emmetropic retina. In the myopic retina, this signal is no longer generated, and this is why myopia doesn't inhibit itself. Yeah, this is said here. And the big question is now, why do myopic retinas no longer use this chromatic aberration business to inhibit eye growth or generate the inhibitory signal? Now, is this also visible in accommodation? These are experiments from the past 3 months or so. Barbara wanted to know whether, or me too, how accommodation responds to these different filtering things. And one thing that we was tested first, subjects look at a red cross or blue cross. Then we use a white cross, but run our filters that simulate chromatic aberrations on these targets. And what came out is, this is known already. If you give emmetropic people a red target to look at, like a red cross on a black screen, they accommodate more. And they have to accommodate more because the chromatic aberration doesn't help them to focus. So they really have to accommodate more. In blue, they accommodate less. But now if you show them these crosses with our filters, then you can see these filters also affect their accommodation. And they also affect accommodation if you take just images and filter them. So basically these James Bond movies. But now if you look at the myopes, they even don't show this response to, when they look at these targets, they accommodate the same in a blue and a red target. So they still, again, don't care about chromatic aberration. So what does this say? I would have loved if this whole business would stay in the retina and not also show up in accommodation, because the big problem is always when accommodation responds to a stimulus, it removes the error signal for the retina. You know, this is this old question. But apparently it does it here also as it did in many other places, but the retina is still getting the signal. And I think this is one of the big unresolved things, how you can remove an error signal by accommodation, but the retina still responds. And I can't tell how exactly this works. This is more work to do. Now, the last, this is only not so long, don't worry. Now we know that inhibitory signal is not generated in myopes. And this is something that industry, optical industry doesn't like to hear. So, and this is probably why nobody cites the paper, because this kind of renders all these approaches with imposed positive defocus to inhibit eye growth in the periphery. This makes not so much sense anymore. And this is a question that I'm interested too. And so let's quickly go through the lenses and what they are doing and what the conclusions might be. The whole business started actually in 2005 when Earl Smith's lab found that you can make monkeys myopic, deprivation myopic, if you still leave the center 24, 37 degrees of the occluder open. So these guys look through it, the fovea gets perfect image, they can normally accommodate. And there's no reason for the eye to grow differently. Only the periphery complains, apparently, that there's no sharp image. And on average, these monkeys get more myopic, even though they had normal foveal vision. This immediately actually was transferred from Earl Smith to the Brian Holden Institute, and they, together with Zeiss, then made these Myovision lenses. These are lenses that just add positive defocus to the periphery. You know all this. The problem with this kind of things is you get a lot of image distortion in the periphery, as you get with progressive addition lenses. When you walk down the stairs with the new progressive addition lenses, it's quite demanding, actually. And this was considered a breakthrough. As you know, there are so many breakthroughs in myopia, but this was a breakthrough. Here it's explained. But if you look at the kids, they were not all that happy. Though these guys, when I first started wearing these newspapers, I didn't quite get used. It's not surprising. The whole periphery is distorted, and they had to relearn how to walk the stairs and up the stairs and things. So this was not a very convenient class. And our first EC project that we had, we got a lot of these spectacles and tried to get adapted. It doesn't really work. At least in our age, it's not working anymore. Okay. And the worst is they didn't really have a big effect. This is some other thing. So this is good for our theory that plus defocus has no effect anymore, because this is what we imposed by these lenses. And the next idea actually came from Jiho To and his student Dennis See. 2006, they had the crazy idea to give this Fresnel type multifocal lenses to the chickens. I never would have done this experiment because it looked crazy to me. But at the end, it was a very powerful experiment, because you can then have two focal planes, one in front of the retina, one behind, and ask, what is the retina doing? Does it take the average of both or not? And it turned out when the contrast is similar in both planes, the retina didn't fade all that much. The chickens became a little bit more hyperopic because its plus defocus has more power than the minus defocus. But then if you go and make the areas that are on plus or minus defocus differently big, so that the contrast was different in two planes, then you could make one plane win against the other, depending on what the ratio was of contrast. This was also working in monkeys, shown by Earl Smith's lab in 2014. And this merged into several contact lens types. You know, this one is very well known. My side is also all these bifocal contact lenses. Now, so they work somehow. But now the next idea was even more funny. Jiho To is a guy who also actually was involved in the development of these multifocal lenses in the beginning. But he was sitting in the bus. He told me that they had in the bus often these plastic shields with the holes in there to make it not so hot in the bus or what. And he said, why don't we add all these little plus lenses in this plane? Then we wouldn't get the image distortions and we still keep the normal spectacle plane. And he got Hoya to make these lenses. You have seen them, the Dean's lenses, and Don has actually one. I brought one that you can look through it. It's not, it looks ugly, but if you look through it, you hardly see anything. This is quite impressive. And the center part of the lens is clear. It's only in the periphery that you get this addition of these plus 3.5 diopter lenses. But you have to keep in mind, these little lens segments have only 1.1 millimeter diameter. The focal length is 28 centimeters. This gives an F number of 260. Nobody would ever build an optics of this kind, because then you can take a pinhole camera. It doesn't make sense. So what comes out of this, even though that the companies all say this is plus defocus that we add and therefore it inhibits eye growth, the image looks very funny that you get through this. I tried to do this a little bit. I have here a camera with a similar optics as a human eye, 16 millimeter. Focal length, F number 5.6 is what we have during daytime or so. And then this camera was focused on a text at 30 centimeters, but then I made the camera presbyopic, basically focusing at infinity, and the text looked like this. It's not unexpected. And I thought now if I would add this DIMS lens that I showed with these little lenslets here, then I should be able to read the text again. And actually, there is a little bit of refocusing by these little lenslets, but the text looks still quite funny. And I think nobody would like reading text of this kind. So it's not really a treatment for presbyopia. This was what I thought in the beginning, but it isn't. Well, after Hoya was quite successful, Essilor jumped in and made a slightly different design, but it's actually the same paradigm. Only they didn't fight with the patents, or they had to fight with the patents, I guess. So this is why they did a different design. And another crazy lens here is the one that you know is this SideClass lenses, which basically impose some diffusion in the periphery. So this is a standard way to induce deprivation myopia, but there was a study showing that these apparently made also myopia inhibition. And you ask yourself, how does this fit? It's not plus defocus, it's basically image degradation, which normally makes myopic. So nobody knows up to now exactly what this is. But I tell you more here. After these lenses sold very well, the first one, the Hoya lens, sells extremely well in Europe. In Switzerland, I'm often in Switzerland, Germany, everybody gets these lenses because, you know, there's no risk. You can't fall down the stairs. There's no risk that you get infections. Vision is perfect. If you look through them, you don't feel disturbed at all. But they work apparently quite nicely in a number of kids, because I have personal contact to some parents and they got these lenses and were extremely happy about it. So yeah. So other companies came in. The Rodenstock came on with a Micron lens just a few months ago, or weeks even. But I don't think, I shouldn't say this, but you know, this is standard positive defocus. I don't know what the sense is. It's already shown that this doesn't really work. You didn't hear this. This goes into the internet anyway, so I get my bomb. This is another design, also plus defocus in the horizontal plane. I have doubts that this works. And Zeiss just came out with another lens, and in this case they had a funny design. They didn't really take circular plus lenses, but these rings that have a strong power in the radial direction, but nothing in the tangential direction. This is 10 diopters in these lenses, and it's also marketed as simultaneous peripheral defocus. This causes nothing with defocus, really. It causes a pretty messy image. And now this is an experiment that I did last week. I was interested in how much do these lenses really degrade a text when you read it. And on the left side, there is just a simple trial lens. So I focus on a text which required a visus of 0.3. You can see the text can be nicely read. Then I add a plus 0.5 diopter lens. It's still possible, but with +1, you can't read it anymore. And to go the other way, minus 0.5, you still can read it, but with -1, you can't read it anymore. So this low amounts of defocus already degrade the image so much that you can't read. And now we can see what these different lenses are doing with the text. So this is this Zeiss lens that I showed you, Myopia. I put this part in front of the camera, or this up and down. And certainly these kind of lens, it's not really a lens, but these structures cause a lot of astigmatism here. And this can be seen on the text. But the key thing here is, even with this 10 diopter stuff that is between the flat pieces of the glass, you can nicely, or not nicely, but you can still read the text. So the effect size of the 10 diopter rings is pretty low. This is now the SideClass, which basically are diffusers. So the spatial frequencies don't change. You can nicely read through them. This is the MyoSmart lens from Hoya, this first from Chiyoto. And if you go through these lenslet arrays, you still can read the text that is requires a visus of 0.3. So all these lenses degrade the image somehow, but it's not more than 0.5 diopters of defocus if you go for reading. And this is a really small amount of image degradation. And the question is now, why does the retina respond so strongly to this? Just to discharge the continuous claim of the industry that you impose positive defocus, I also did the following simple experiment. I again have here a text, then I take a defocus of -10 diopters and add this Myocare lens that has +10 diopters of defocus in these rings. You can see there is no defocus or no refocus, which means this lens doesn't really cause positive defocus at all. That can be compensated with a -10 lens. And this is true for the other lenses. Basically, my conclusion is at the end, there's not really any positive defocus involved in all these lens designs. Basically, they degrade the image in a funny and complicated way, and the retina doesn't know what to do with it, but it apparently triggers the positive or the inhibitory signal for eye growth. And so we have to look for some image degradation that is efficient, but it has not to do with positive defocus in my observation. And this is my conclusion too, and I'm then done. So the induced peripheral image blur is surprisingly small. This was striking. It's about as you would defocus by 0.5 diopters. This is very different from the old Zeiss lenses, which really imposed quite a bit, but they didn't work. Then the lenses, this is my key conclusion, do not really impose positive defocus, even though it's advertisement argument all the time. It just makes funny image scrambling, but not positive defocus. And I mean, this other kinds of image degradation might be the reason why they work also in myopes, which don't respond to positive defocus. As you know, in the US, you don't get any of these lenses up to now. And as far as I know, it's also not planned in the near future. But in Europe and Asia, particularly the Hoya lens is extremely well accepted. Okay, this was my talk. Thank you very much.
Question in the chat?
No, that was just reminding everyone.
Okay, so hopefully if you want to read them to me if they come up, or how do you want to do that? Okay. Any questions in the room before we worry about what the chatters are doing?
So you talked about the fact that it's not positive defocus, that it seems to be image degradation, but you talked about the SideClass lenses as though they were kind of funny. And it seems like that would be an image degradation type.
It's a
thing. How would
contrast.
that differentiate?
Contrast reduction is only contrast reduction. I think the spatial frequency spectrum and the alignment of the pixels is preserved. This is maybe a different mechanism. I can't really tell whether it feeds into the other mechanisms, because contrast reduction is certainly involved in the other classes as well. One thing that one could argue is that if you have on-off system and the imbalance between on and off is critical for emmetropization, you can say, if you get everything diffused or low pass, not low pass filter, diffused with low contrast, then also the difference between on and off receptive field responses is reduced, you know. And this is something one has to test. I think nobody knows why they work actually, or if they really work. There's a 1-year study, I think, but I didn't hear more than this.
What are your thoughts? Where do your thoughts lead you if you believe this chromatic comparison is important in unlocking an inhibitory mechanism?
Yeah.
What's a way that you can have you thought of implementing this? I mean, if
actually what you could do, and what we are testing this right now, is you could certainly use these filters on a regular computer screen. So the screen is now a low pass filter in the blue, and the effects are not very strong. You can reasonably well work. It's not very strong for your subjective sharpness, fortunately, because if you would have to filter the red to get this result, this would be bad. But the blue works, and so you could implement it in any computer screen. And what you have to do.
Picture this as being something that's done over long periods of time, or do you think it would be powerful, like say the red light therapy seems to be?
Yeah, the red light therapy is something. This is an extreme case, whatever it is. It's an extreme case. We can discuss this separately, maybe. But I think if you look at these DIMS lenses and also these SLR lenses, there were studies published actually where they gave them to the children only 1 hour a day or 2 hours a day. And the big effect sizes were actually between 1 and 2 hours. And then when they wear them all day long, there was a little improvement, but the major effect was for a pretty short period. So I could imagine that you don't have to do this all day. And if you do it an hour or two on the computer, it could be that there's already a long-term effect. Jeff?
This is quick. Do you yet have a publication that indicates that it's likely not positive defocus that causes myopia, owing to myopia progression, but it is image degradation?
Actually, yeah, I can say I'm writing or wrote a review for Progress in Retina and Eye Research to put all these things together. And this one was reviewed and I revised it, but I think it will be going out. Yeah.
Okay, so look for it soon. Anybody out there in TV land? Okay. All right. Well, I need to present Frank with his Hill medal. I think it's a medal. Let's see. Yes, indeed.
Oh, wonderful.
And so, Sarah, will you do, if we walk over, we'll do pictures. We'll let everybody go and then we'll do pictures.
Oh,
so
quite
that's
impressive.
Commemorating your
thank you very much.
And certainly thank you to Dr. and Mrs. Hill for endowing this lecture during the pandemic. We had speakers from around the world as well, virtual, but it's lovely to be back in person and to have the Hills endow this at a level that allows us to bring someone of Dr. Schaeffel's stature. So thank you all for giving us a chunk of your evening, and I'm so glad you were all able to attend. Thank you.
Thank you very much.